I'd like to go into our question and answer period, if you all are ready. All right, so we have a string of questions from some of our attendees. The first one goes to Dr. Overton. Thanks for sharing this information. The SARA results show weathering over a time scale of years. I'm having a hard time reconciling these results with the slide explaining the weathering of C1 to C25 compounds. Did the SARA analysis include C10 to C24 compounds?
Good question. The weathering process, depending on the oil and where it was, took from days to, if the oil got sequestered on shoreline, weeks to months to years, right? So some of that analysis, some of that oil had been on the shoreline and weathered. So depending on how the oil is mixed in the environment with the water, with the emulsions, it weathers at different speeds, right? So that particular paper was looking at, you started off with the oil that floated on the surface, and then it looked at particles of the tar balls and the surface residue balls that were collected on the beaches, right? So it was looking, and of course that process was over a couple of years when they were studying this. So if the oil had not, if they had collected oil that was floating offshore, you would have gotten a much different picture in the SARA analysis that was done and described in that paper, right? So again, the gaseous components are lost from the petroleum very, very quickly. The C10 to C24, I call it C10 to 24, 25, they are typically lost if there's well mixing of the oil in days to weeks, and the heavier components, the C25 and up, take much longer. Joel showed pictures of them being buried on the beach, and the color lasts for a year or two, right? We've got areas in the coastal marsh where this material was deposited into the marsh, and I can still go down there today and pick up a clump of that material that has not been weathered over that time. Notice that Joel showed pictures of bulldozers and mechanical equipment, which you can put on a beach. Unfortunately, when you go into a marsh, you're talking about a very, not a very solid footing, and you actually walk on the marsh, you'll sink down in it. So we can't put heavy equipment on. In fact, you almost can't get this material out of the marsh. So the material that went in the marsh, some of it is weathering. It's weathering slowly, and it is still there today. Does that answer your question?
I think that covers that person's question. A more general question for you all, how much oil naturally seeps into the Gulf of Mexico each year?
Boy, that's been a pretty hotly debated question. Mineral Management Service, some of the people of that agency have studied this. I have heard numbers on the order of 20 to 50 million gallons a year, depending on who you talk to. Remember, there was 200 million gallons a year dumped in a short period of time from the Macondo spill. So there is a lot of oil over the entire Gulf. The Macondo spill was focused in a fairly small area in the northern Gulf. So, but there is a lot of oil, and as Joe pointed out, the bacterial community in the Gulf is used to having natural oil seeps and spills, and it's well acclimated with the molecular capability to degrade the oil.
Okay. Before we go on to the next question, I just wanted to point out to you all that there is an evaluation link for this webinar available in the chat box. So if you can go to that at some point after this webinar and give some feedback to help shape future events that we do, that would be great. Moving on to the next questions that we have. Dr. Kostka, can you please confirm that your comment that most oil breaks down by degradation is specific to the case of oil deposition on a sandy beach like Pensacola? Also, how would the rate of observed biodegradation change for a colder climate like the state of Washington? And lastly, what happened to the tar balls? I
So
can repeat any of that if you want.
great question. So I want to thank everybody for participating. I saw almost 100 people participating at one point, and now we still have almost 80. So thanks for hanging on. So question about biodegradation versus other pathways for degradation, that, like, for example, photochemical degradation that Collin talked about. And my first point would be it's going to be totally dependent on the environmental conditions. So my work is mostly in soils and sediments, so the benthic environments. And so in those environments, a lot of times there is no sunlight, right? Because these oil particles are buried below the surface. So in that case, biodegradation is going to be more important, certainly, than photochemical degradation. So keep that in mind. Also keep in mind that the oil that reached the Pensacola beach would have been already impacted by chemical weathering and by the processes that Collin mentioned. So that stuff was that oil was rising to the surface in slicks and then eventually making its way to the coastline where it was beached. At least in our hands, what we found was a lot of the at least medium size hydrocarbon compounds survived that chemical weathering process. So we still had a fairly large concentration of alkanes in the oil that reached Pensacola Beach. And then, as I showed, a lot of that oil was degraded in 1 year's time. So what are some environmental conditions that are important? At least in terms of biodegradation, microbes like warm, moist environments with plenty of oxygen and nutrients, as I mentioned. The beach satisfies all those requirements, with the exception that it's a kind of a nutrient poor environment. So for those reasons, we think oil biodegradation was really important. We were concerned that perhaps since the beach is such a dynamic environment, that some oil could be washed back out to sea. But when we did experiments in the laboratory using that big aquarium, that mesocosm setup that I showed you, we found that almost all that oil was biodegraded. And so we got similar results to what we found in the field. So we think that the majority of oil in that case was biodegraded. Okay, so I've gone on long enough about that. So how would it be related to biodegradation in a colder climate? And one thing I didn't mention is whether that biodegradation is occurring in the water, in seawater, and the water column and sediments, as I've just been talking about, is really important. We have seen that temperature is not as important as we thought. In other words, oil degrades pretty fast under cold conditions. There have been several studies, at least in seawater, that have shown that biodegradation occurs pretty fast in those conditions. There's a lot less information, though, on biodegradation in cold conditions. So I'll make a plea, just as Collin did, for more research to figure that out. What happened to the tar balls? We did an in situ study. So basically, we put tar balls inside tea balls, inside stainless steel tea containers, and we put them out into the beach. And what we found was that basically they went away. So the mass of those tar balls went away over a 3-year period, and we could see that was slower than what we found for smaller oil particles, but indeed that the tar balls were degraded. So the sand that makes up that tar ball is going to stay there, right? That's not going to go away, but the oil part of that tar ball will be degraded and forming carbon dioxide and what I said, and probably a lot of intermediate compounds too.
All right. Our next question: if oil-eating bacteria takes over microbial communities when oil is present, are there observed consequences to ecosystem dynamics?
So also a very good question, one I can answer in a shorter period of time, hopefully. So I tried to talk about this at the end. I didn't have a lot of time to talk about it. So using microbial genomics, we're very good at figuring out what types of microbes or genes or biochemical pathways are present. We're less good at actually seeing what effects oil has on how ecosystems work, on their functions. With that said, we were able to show some very clear effects on nutrient cycling. That is the way that microbes process nitrogen. And we saw that very specific ecosystem functions, this is work that we're just publishing now, what we call and denitrification, so nitrogen removal from the ecosystem, is very much affected by oil, and those processes are actually poisoned by oil to some extent. So yes, we saw, at least in the beach sands that we study, that there are ecosystem consequences.
All right, Joel, are you okay with another question?
Sure, bring it on.
All right, how are oil degrading microbe taxa in beach sands compared to salt marshes?
Yeah, and I see Irv Mendelzone asked this question. I hope Irv is still on. Thanks for the question, Irv. Good question. So Ed touched on this earlier, and I said, as I said before, biodegradation is going to be very important. Is going to be determined by the environment. And one way that the salt marsh soils are very different than beach sands is that they go anoxic. That is, oxygen is consumed very close to the soil surface in salt marshes because they're muds, and oxygen diffuses only very slowly into the soil. And so only oxygen that's mixed into the soil by burrowing animals or by plants is going to get down into that soil. So what I would say is that the microbes will be different in the sense that the salt marsh soils are anoxic, and so you're going to have a predominance of anaerobic microbes, at least below the top millimeters to centimeters in the soil. Now, with that said, with the studies that were done on microbes and what I've seen, we didn't not study salt marshes. We've studied salt marshes, but not oiled salt marshes. So I would expect you get similar aerobes, so microbes that love oxygen at the soil surface. And then as you go down in the salt marsh soils, you'll have these anaerobes or oil-eating microbes that like no oxygen conditions. We did do one study on those anaerobes in sediments that were anoxic, shallow Gulf of Mexico sediments. And we found sulfur bacteria, sulfate-reducing bacteria that degrade oil under anaerobic conditions. And I will just say too that we are only scratching the surface on the types of microbes that are out there that even do aerobic respiration. We can't grow most of the microbes in nature. And when we've just published what's called the Genome Repository for oil for oiled systems, and we found 2,000 new species of microbes that degrade oil using metagenomic techniques, and only less than 5% of those microbes have actually been grown in the lab. So we don't know about most of the microbes that are out there. We need more research for that. We know a lot more about the aerobes. We know less about the anaerobes. I can tell you that some of the anaerobes that have been identified in salt marshes that degrade oil are known to science, and so we can point to certain sulfate reducers, for example, that degraded oil and antigens, and some aerobes were similar.
Joe, could I add a little bit to that?
Sure.
The difference in the physical energy that the oil on a beach experiences and oil in a marsh. Basically, anybody that goes swimming along the Gulf Coast sees the wave action, and it's really turbulent. It's mixing oxygenated air down into a porous material, which is the beach sand. That's a completely different situation with oil on the marsh. You don't get this massive physical alteration every year. So the oil kind of stays globbed together as a solid as opposed to these smaller particles. And so I believe that that the biggest problem is that you just can't get in and clean up a coastal marsh very effectively. So whatever's there stays there and globs together, and then it's very, very, very slow to degrade, either aerobic or anaerobic. We know that asphalt roads are aerobic in an aerobic environment, and they don't degrade. So in addition to the microbial and the oxygen species, it's the solid material. Once it gets that solid material, it is really, really very difficult to degrade further.
Yeah, just to follow up with that a little bit and say that would be one of our recommendations for emergency response, is to focus on salt marshes more so than beaches, because the beaches, as I presented, you're going to have more rapid biodegradation. and you can observe the oil there, whereas in salt marshes, you have muddy sediments, oil is going to degrade much more slowly. I actually have studied oiled salt marshes since before the Deepwater Horizon oil spill, and you can go back there decades later and find basically liquid oil 10, 20 centimeters down in the soil. So it degrades very slowly, and for the reasons that Ed mentioned. And I would add one reason, and that is that the microbes just have less access to the oil. If the oil is present as a liquid, or if it's present as larger particles or droplets, there's simply less surface area available for attack by the microbes. So it's what form, physical form that oil is going to very much affect biodegradation, along with what Ed said.
All right. Thank you for that very thorough answer, guys. Going on to the next question in line, is there any hyperspectral imaging data for natural oil seeps?
I think you're talking to the wrong people. At least I'm the wrong person. I'm sure there are satellite pictures that show seeps. Ian McDonald is the guy to go to with for that information. Collin showed some of his data that was taken from satellite imagery, I'm sure.
Yeah, the data I showed with the oil moving around the sea surface, that's satellite aperture radar data. But there's lots of remote sensing tools being used to track oil movement at the sea surface right now, and lots being developed. So,
Okay, great. Our next question is a little bit more of a comment, maybe for you all to comment on, or maybe Ed more specifically. SARA analyses themselves can be biased, as the light ends are normally lost as a function of the analytical process. Be careful with SARA analyses. What are you all thinking about this?
Well, SARA analysis is really not designed for petroleum that comes out of the ground. It's after the light ends have been separated from the liquid oil. So it's what the chemists use when they take the oil to a refinery and decide how to operate the refinery with that material, right? So it's not, SARA analysis simply doesn't detect the really light ends. Now, we were dealing with a spill of a material that was petroleum, came out of the ground as petroleum, and we had a very massive amount of natural gas as part of that material, right? That natural gas and the volatile components were separated fairly quickly. Almost none of the samples that were collected from the ocean surface, for example, had alkanes below about C13 or 14. So after the oil got to the surface, things more volatile than C13 or 14 had been evaporated off. What floated and came to the shore had alkanes in the general range of C16 to 17 and higher. I don't know whether that answers the question, but clearly you've got to use the right analytical technique, and no one single technique analyzes the gaseous components of oil, the VOCs, the volatile components, the semi-volatile components, and the insoluble high molecular weight asphaltenic fractions, which require, of course, that high resolution mass spec technique.
Okay, great.
can I just add to that? I think one thing that maybe we could stress is what Ed just said, this concept that no one technique is going to be a silver bullet. Whenever you're dealing with complex mixtures, you always run into these situations where you only see what your analytical tool can see. And a lot of these analytical tools we have are blind to certain types of compounds. So the way that we combat that is by piecing together multiple analytical tools to attack the same question. And what we found for a lot of the research over 10 years of studying this problem is that from a chemistry perspective, they're all pointing towards similar answers. And that makes me really comfortable with some of the things that I say about the way the oil gets processed when it's floating at the sea surface, when you have multiple chemical tools that all tell you the same answer. So, you never want to rely on one tool. You always have to sort of bring a Swiss Army knife and go from there.
All right, that's an excellent point to bring up. We have time for maybe just a couple more questions. It's actually 3:30, which is when our webinar is supposed to end. Do you all mind answering a couple more questions, or what's your preference?
We're here.
On the spot. All right. So since high molecular weight oil in an anaerobic environment weathers very slowly, what happens when disturbed, when a storm aerates the oil? Does it become available to multi-cell organisms and potentially create physiological harm?
So I'll start out with this, and then I'll let my colleagues follow up. So this is something that definitely happened and continues to happen in the Gulf of Mexico. I talked about beach sands where you can see the oil on the shoreline. I didn't talk about all of those soils and sediments that are buried by water, right, that are in subtidal environments that we can't see. And it was really hard. This is actually one thing that I tried to do in my science, was to track oil in subtidal environments at the seafloor, and it's really hard to do. And we continue to see oil being resuspended and released from storm events and washing ashore. And the answer is yes. If it was buried in the soil, it's likely not degraded, and it will still contain a lot of compounds, like Ed was mentioning, that are potential carcinogens. So it can have an effect on multicellular organisms and potentially create harm.
Okay.
Joe, I was pretty impressed by the fact that these heavy molecular weights generally have the dark color, and the color went away in some of your sands and this is below the surface, so it's not photochemical processes down there. Yeah. It's anaerobes and aerobes that are degrading that oil. Unfortunately, that doesn't happen in the marsh, because the marsh is anaerobic just below the surface, and then you've got what you laid on top of the surface, which was generally almost a mat. And so it was a kind of a crusty mat that was laid on top. There was a little bit of oil under that mat, some of it trapped in fiddler crab burrows, that was pretty liquid, and that material, some of that material is still down there. And if you go in the right spot, you can see it sheening on with wave action, and you can actually collect a little bit of it. Nothing like what we saw in the summer of 2010. Let me emphasize that we are a long, long way from June 2010, but there is still oil residues that can be found, particularly in the coastal marshes that were impacted.
All right. If no one has anything else to add, we're going to go to the final question, which deals with the Taylor Energy spill. I'm curious about the Taylor oil spill ongoing since 2004. Taylor Energy have long decommissioned the site, but contend that nothing else can be done to contend the spill. They claim it's no longer active and that plumes are from contaminated sediments. What are your thoughts?
I've worked on that spill a little bit. First, let me say that to compare the Taylor spill with the Deepwater Horizon spill is just absurd. I mean, there was massive amount for 87 days, I believe, of fluid flowing into the Gulf of Mexico. Taylor is, you're talking maybe 100 gallons, I think, a day or something like that. So rather than thousands and millions of gallons a day. So it is a sheen. It is kind of akin to natural sheens, although this is from a non-natural event. That well was collapsed by a mudslide caused by Hurricane Ivan. A lot of oil is retained as a solid, a semi-solid viscous mass buried in the mud down there, and some of that is leaking. Whether there's a pipe that is attached to a reservoir that's still flowing is causing the leaks, or whether there's natural gas bubbling through it is the real question. The good news about all of that is it doesn't seem to be causing detectable environmental impacts. What I mean by that is if you go down and look at the beaches, I don't believe there has been a single sample of Taylor oil, which can be easily fingerprinted, but no sample of tail of oil has been detected in the beaches along the foot of the fingerprint, the foot, the bird's foot area of the Gulf. So the good news is that we're not looking at an environmental disaster. The bad news is that it is a persistent a leak very similar to Sheen's and other parts of the Gulf. And the question is, what do you do about it? Is there something that can be done? If you go down and interfere with the oil on the bottom, you're going to disrupt oil that is entrapped, and you're going to have a much bigger release over a shorter period of time. And we do know that that's what we don't want. We don't want a rapid release. Mother Nature can take care of it. So the next question is, what do you do about it? Do you just let it sit and hold your nose and say this shouldn't have happened, but it's what we got? Or do we go down there and try mitigation areas? Currently, the Coast Guard is trying to put a dome over it and collect it, and I think they're being fairly effective at that process, but they're still sheening even with the collection dome in place.
Thank you for sharing that, Ed. We have one last question, which I know that I said the last question was the last question, but I think that this is good perspective for everyone who's online. We have a question about how is all this combined research best passed along to those who will determine how to better respond to the next Macondo. And I know that all of you are part of the Genome Synthesis effort for Core 2. So I was wondering if you all could very briefly share the synthesis effort that you're working toward.
Well, let me start out real quickly by saying that after most oil spills prior to the Gomery spill, the Deepwater Horizon spill, the research was really intense during the spill when it was with high public interest, like the Exxon Valdez or the Ixtoc spill in the southern Gulf of Mexico. A year later, the research funding dried up. With the Deepwater Horizon spill, we've had 10 years of very intense $500 million. And there's actually more money than that because BP put two other stanches of $500 million into the NSF and to the National Academies. So there's a lot of research going on, both in the environment and into the communities that were affected. So we are learning a lot. Collin's example of the effects of photochemistry is a perfect example. We're learning a lot more about the biota and the ecology in the deep Gulf, which we knew almost nothing about. This was a surprising spill. For example, we didn't know, we assumed that an oil spill in the deep ocean, most of the oil is less buoyant than water would come to the surface. We didn't know there was going to be a subsurface plume of dispersed oil and natural gas. So that there's a lot that we have learned about this spill. Joel has pointed out all of the ways that the microbes and the genes of the microbes are degrading oil. We've learned an incredible amount, and that information is passed on through scientific publications and our scientific meetings to people that are involved in spill response.
So I'll just follow up on that a little bit. The fact that Ed said we've learned a lot. So one of the first Gulf of Mexico meetings I went to, they were counting dolphins in Pensacola Bay. And after the guy finished his talk, I said, "Why are you counting dolphins in Pensacola Bay?" And he said, "Because we never knew how many dolphins there were in Pensacola Bay. So we don't know if the oil spill had any effect because we never had any data before." So I think that's just illustrative of the fact that we have baseline data after 10 years of research to understand, for example, how the fisheries were impacted by oil. So that's something we could bring that information to bear to figure out how the ongoing Taylor spill might impact environments there. More to the beach environments that we study, for example, and as I said before, I think one way that this information can already help us is to prioritize sites for emergency response. We've learned that we can't clean up oil from salt marshes. You know, I don't know if you've seen photographs of responders trying to clean up oil. They pressure washed the marsh. They used like baby diaper type of material to try to take the oil off of the blades of grass in the marsh. None of these methods were very effective at all. In fact, they did more harm than good in the salt marsh. So that's one thing we learned, was that we need to prioritize salt marshes during emergency response. So we've already learned some information that we can use. In terms of the microbial information, I would say two things. For one thing, it's just helped us to get enough microbial genomics information about the environment so that we can reach the point of where we can use it. I mentioned the Genome Repository, so where we've now collected information from thousands of new species of microbes from the Gulf of Mexico. We think the Gulf of Mexico is prime for oil degradation, but really we don't know, because now we have more microbial genomic information from the Gulf of Mexico than we do from a lot of other ocean environments. So now we need to do more comparisons to other oceans that might be less primed for oil degradation. So we have a lot more information that we could use. I could say two things specifically about how microbial genomics could be used. One is as indicators of the state of oil, of oil contamination that's present in the environment. So use a microbial bioindicator that might be easier than taking a sample and running a mass spectrometer, running it through a mass spectrometer to figure out what oil compounds are present. And then also using genes as an index of degradation to prioritize, emergency response.
I will say also, I think those are excellent points. In response to the question as well about how that information is passed along, our Sea Grant program, the Oil Spill Science Outreach Program, we've worked very closely with the emergency response community since our team has been formed. And so we've worked with them to create workshops to share GOMRI and other science with them, as well as try to create those bridges between academics and oil spill responders so that hopefully things can be increasingly more fluid over time between those two worlds. Do we have any more comments from our panelists?
Maybe I'll add something about the synthesis effort, because I think it is important to point out. So GOMRI put up a quite sizable amount of money over the past couple of years to piece together teams to target a comprehensive synthesis of different topics. And there are several examples on the table here. Ed talked about analytical chemistry. I talked about photochemistry. Joel talked about microbiology. Bringing together teams that are working on similar questions, but forcing them basically to come to reach some consensus about what we've learned, and then documenting that in ways that can be understood by non-specialists. So there's been lots of documents that have come out that are written for the technical audience, but then some documents that are written for non-specialists. And that's been very important to sort of translate these ideas and these findings to more actual useful information for responders. And the second thing I'll add to that is Ed mentioned this cyclical nature of the funding cycle of oil spill science. And I think that this synthesis effort might help bridge that gap. I think one thing I learned from the synthesis effort is there was just a lot of research that was buried in the literature that was cutting edge 30, 40 years ago and never saw the light of day, but would have been helpful to responders if they had access to it. And I think part of that is driven by this cyclical nature where people go in and out and working on oil spill science. And GOMRI gave everyone an opportunity to work for a full decade on this topic, which is a rare opportunity. So I think that in and of itself is going to change how this research that we're doing is translated to actual relevant science for first responders.
I think that's an excellent point. With that, I would like to wrap up and say thank you to all of our guest speakers. It was wonderful to learn from each of you, and I really appreciate you all sticking around for all of the questions, and to all of our attendees who've hung in there. Again, please check out that evaluation link. We actually really do read all of the comments and try to use that to better inform our future events. And also recordings of this webinar will be available on our webpage gulfseagrant.org/oil spill outreach. So thank you so much to everyone.